Hi, good morning, folks. So today we were talking about how to use the phylogenetic trees. So it sounds like in one video to use the phylogenetic tree and understand all the software, it kind of criminals, but I'll do it part by part to show you without getting lost in the world of reuse phylogenetic tree from you get the raw sequence until you visualization of the trees. So let's talk about the free software you can do, and I actually use a couple of free software to do the phylogenetic trees, except one thing. I have license at my lab works. They have license for Genius Primes, but actually I use Genius Prime to do the quality checks of the raw sequence, and then do multiple alignment and do the blast to find out the species like that. So the first software is called MrBayes. Here is MrBayes. So you might want to Google or any browser you like in your computers. This is not a dark web, by the way. So this is MrBayes website. So basically, when you type MrBayes, you will see a couple of the MrBayes program websites. And if you use Genius Primes, I mean when you have the license, so you have the MrBayes plugins, you don't need to download the files. So within their website, they have a brief fleet of the MrBayes. So MrBayes is the program for Bayesian inference and model choice across the wide range of phylogenetics and evolutionary models. MrBayes uses Markov chain Monte Carlos, which means in short MCMC's methods, to estimate the posterior probability distribution of models or parameter. So instead of using default in MEGA to be used phylogenetic trees, we use MCMC and generation maybe up to 1 million. So we're talking about that later, but now to install and understand their interface. So you might want to click on download under the picture of MrBayes, I think. So you download it, and what you're looking at is that the MrBayes 3.2.7 zip files. If you use zip files, then you start download. When you extract the file, you have this couple of batch file right here. I mean, readme. It's a description of MrBayes and what you want to read about the MrBayes itself, the manual tutorials. You might want to see in the docs file. You see the manual MrBayes. So right here, I think you have to read through. I mean, to know which model you use, which command line you want to start writing, because MrBayes is the command line you type in, you hit enter, they run. So basically, like TR Studio, you have to type everything in instead of clicking the option they have like in Megas or SPS. So a simple analysis, getting data in MrBayes, checking the model, setting up the analysis. So you may want to read that, but I show you the bypass way by using the other softwares called MEGA. So after you download it, the file of MEGA, the file of the MrBayes in the bin folder failed to Win 30 and 64. So we basically use 64. So it's a trick right here. When you download the MrBayes, it will run directly in your directory path file locations. It's like you set the set WD in our studio codes. However, you can bypass that by copy the files into the folder where you have stored the data. So I will show you right away from here. So this is my folder of data. So I have a FASTA file, Philip and Nexus file, and MrBayes. I just copy the MrBayes from here to here directories. So when I click on and open the MrBayes, it will run only in this folder. So if you have many data sets, many a location file, you just simply copy that MrBayes file directly into the folder. So by the way, MrBayes reads the Nexus file, so you might want to export your FASTA files to Nexus file in order to read the file in MrBayes. So another software is called MEGA. You want to download it. So MEGA right here, here is the software with the green logos. MEGA software, that's something that related to the sequence alignment and DNA nucleotide, something like that. So you might want to click on MEGA software, and then they will ask you which do you use, Windows, Mac, Ubuntu. So right here they're choosing the right method of the my computer. So I click on download. You have to fill out your some information to download the MEGA, in case they didn't let you download. So I'm from Cambodia, I'm students, and then I am a university student. And then the institutions, I just taps my university name, so I click download. But I actually already downloaded, so I hope you guys know how to install your files application in your computer. So we were not talking about that. Right here we're talking about MrBayes a little bit before we move on to the next chapters of the videos. So MrBayes itself, when you open it like that, so MrBayes is showing only these black screens. I'm not racist, but it actually is. So Bayesian analysis of phylogenetic is distributed under the GNU General Public License. To know the abbreviation words of the command line you want to set up the models or parameter, you might want to tap HAP and then enter. So the command line right here, you will see a lot, but we will focus on some of the command lines. So MC is start Morco chain Monday color analysis. We will use that command line, and then I think LSets, we set the parameter of the likelihood models, basically the models. Another things that you can read your file from here. I will not go deeper in this video just to understand it. So the next video we'll be talking about more complicated than everything, but don't feel lost in this video, just install and understand the software. So it's a free software. So here is the file names. Like I mentioned that MrBayes reads Nexus files, so .nex means Nexus files. You might want to rename, click on F2 and then copy the name. You open the file in MrBayes, we using execute, and then put it inside the double quote, .next outside double quote, semicolon, click enter. So you have successfully reached your files. I have 80 sections. They briefly your file. So we successfully read the files. Another one is that talking about the MEGA. So MEGA right here is so right here you have models, find best DNA protein models, and then they will lead you to your location files, where I have stored a file is 80 document alignment, and then my data is nucleotide sequences, not protein. Protein coding, yes, then I use the standard one. By the way, I use CO1 for the primer, by the way, and then I keep it default right here, click on OK, and then the MEGA will calculate the number and the best model for you to calculate and view the phylogenetic trees. So what you're looking at is the bunch of numbers. It's a bunch of bullshit. Anyway, the model you might want to use in MrBayes is GTR plus G plus I. The parameters is 167, and the BIC, I don't know, I forgot it. What does that mean? I forgot it. So the models right here, GTR plus G plus I, in abbreviation right here, GTR means general time reversible. So we will use general time reversible, and then gamma, and then invariant size, something like that. I don't know, I forgot it. So this result can save as the Excel files. So I hope and I suggest that you save this in Excel file to defend yourself when you open and ask why are you doing this, and then you pulling up the Excel file, say it's the best model that determined suggestion by the Mega. Alright, so I already saved it. I didn't save it twice. Alright, so one thing that's before we saying goodbye to Mega, just want to tell you what GTR plus G plus I mean and why are they important to do. So right here, you might want to click on Phylogenetic to produce the phylogenetic trees, construct test maximum likelihood trees, and then use the active data. So right here, the interface to produce the phylogenetic tree in Mega. So right here, if it comes with the default, it might be say this number is 500 bootstrap replicates. So I suggest you to go with 1000 bootstrap replicates. It's a common use by many scientists to produce the phylogenetic trees. And right here, I guess the default one might be not general time reversible models, but I just changed to show you guys. So the models are measured right here. We use the GTR right here, and then right alongside we use gamma distributed with invariant sign min plus G plus I. So in short, GTR plus G plus I, and then the gamma, the categories we use files, and then keeping it default. So and when you click OK, the Mega will produce the phylogenetic tree. So it will time approximately 1.5 hours depending on your computer specs. So it will running from 3, 4, 5, 6, 7 until 1000 bootstrap replicate numbers, and to produce the phylogenetic tree. By the way, I'm not producing it in the Mega, so I'm gonna stop. Yes, and the instance phylogenetic tree that they will use. Yeah, it's so let's move on. Coming back to MrBayes. This video showing you how to download MrBayes and Mega and how to determine your sequence model to produce a phylogenetic tree, and some command line to read successfully your file in MrBayes. So before jumping to this part, you might want to use the other softwares like Genius Primes or Pro Eugene. I don't know, maybe I forgot about it. I don't use it, but they actually a free software to edit the raw sequence, to check quality, to cut out the primary regions, and then do the pairwise alignments, to blast, find out the spacing, and then do multiple alignment to get this file. So to get this file, it's spending a lot of time to complete. So alright, we'll see you guys in the next video talking about how to produce the phylogenetic trees again.